Hello dear students, welcome to all of you. So in our previous lectures we have started new unit, that is milk chemistry and constituents. In our first lecture we have covered definition and basic components of milk. In second lecture we have started detailed component analysis of milk. In that we have studied milk proteins. So in this particular lecture we are going to study enzymes in milk, that which type of enzymes present in milk and their role they play in milk and their importance as a constituents of milk. So you all are familiar with the term enzyme. So enzymes are biocatalysts. They are synthesized by living cells, and why we call them biocatalysts, because they speed up the rate of reaction without altering themselves, and as they are synthesized in living cells, that's why they are known as biocatalyst. So in milk also there are several enzymes are present, and they play a very important role in milk, as well as they are a lot of importance as far as dietary components are concerned. So enzymes, they are made up of, all enzymes they are of made up of proteins. Okay, enzymes mostly present in the form of colloids or colloidal suspension. There are different types of enzymes. Okay, so basically they are classified according to the chemical changes they bring about, such as hydrolases. These are also known as hydrolysing enzymes. So these enzymes break down complex molecules into simple form. Then oxidases as well as reductases. So these are enzymes. So these enzymes, they are basically involved in oxidation reduction reaction. The enzymes, they are also classified on the type of substrate they attack. For example, proteases. So these are nothing but protein splitting enzymes. So basically these enzymes act on protein, and they break down protein into polypeptides, or again further they break down these polypeptides into simple amino acids. Then lipase. So lipase are fat splitting enzymes. So fat splitting enzymes. So the lipases, they break down fat molecules. Okay. Then amylase. So amylases, basically they act on different types of carbohydrates. Okay, and after breaking the carbohydrate, we'll get sugars in free form. So amylases basically act on different types of carbohydrates. Enzymes, we know that they are very susceptible or very sensitive to physical factors like pH, temperature, then heat. Okay, so their activity mostly depend upon these factors. Okay. Now in case of milk, enzymes in milk, they are occur in 4 phases. Okay, so they are water soluble, casein bound, lipid bound, and some of the enzymes, they are present in microsomal particles. Okay, so in milk, if you see, there are some enzymes, they are soluble in water, and they are categorized under water soluble category. In some of the enzymes, they are bound to protein casein, and some of the enzymes, they are also found to be bound to lipid. Then there are some enzymes, they are found as a microsomal particles. So these enzymes are in state of equilibrium in milk. Okay, so all these 3 to 4 stages, they are equilibrium with each other, and these 4 phases, or these states, they can be changed by due to agitation, homogenization, as well as heating. Okay. So first we will see peroxidase. So peroxidase enzyme liberates oxygen from hydrogen peroxide. So peroxidase is an enzyme that acts on hydrogen peroxide. So peroxidase breaks down hydrogen peroxide, and after breaking hydrogen peroxide, oxygen molecule get liberated. So basically in milk, in fresh milk, peroxidase is present in the form of lactoperoxidase. So in fresh milk, the peroxidase that is present is known as Lactoperoxidase enzyme. So activation of lactoperoxidase result in enhancing the keeping quality of raw milk for longer time. Okay, so raw milk, if it is left for longer time, then definitely the peroxidase get activated in that particular milk. So peroxidase basically works for raw milk to kept for longer period. It is destroyed at temperature above 80 degrees Celsius. Okay, so this enzyme destroyed after heating milk beyond 80 degree Celsius. The next enzyme is amylase, that is also known as diastase. So amylases, you all know that amylases are the enzymes that acts on different types of carbohydrates, and they bring about breakdown of carbohydrates into simple sugars. So in milk also amylases are present, and this enzyme hydrolyzes starch to dextrin, and it is present in milk in very small amount, and it is destroyed at 60 to 65 degree Celsius. So amylase is present in milk in very small amount or very trace amount. Then lipase. So again this is hydrolytic enzyme. As you know, lipase it acts on lipids or fats, so it breaks down fat molecules. So it hydrolyzes fat into corresponding fatty acids and glycerol. It causes butyric or hydrochloric acidity in milk, cream, butter, ghee, and enhance acidity. So basically lipase is responsible for acidity in milk, cream, butter, ghee, okay, and also it enhances acidity in milk. So this particular enzyme lipase, it get destroyed after heating milk at 63 degrees Celsius for 20 minutes. Okay. The next enzyme, it is catalase. So catalase decomposes hydrogen peroxide into oxygen and water. Okay. So catalase basically is an enzyme that acts upon hydrogen peroxide, and it breaks down hydrogen peroxide molecule into oxygen as well as water. So it is present in small amount in milk. Okay. So on contamination of milk, its concentration increases in milk. Okay. So if milk get contaminated, then catalase concentration get increased, and it is destroyed when heated above 90 degrees Celsius for 20 to 25 minutes. Okay. The next enzyme is phosphatase. So this enzyme basically hydrolyzes phosphoric acid esters. During pasteurization of milk, this enzyme get destroyed, and this enzyme is thus used as marker to check proper pasteurization of milk and milk product. So this is very important. So if you want to perform pasteurization process, then this particular phosphatase test is an indicator method that milk is pasteurized or not. So this particular enzyme phosphatase is get destroyed after heating at pasteurization temperature, and basically what it does, it break down enzyme phosphatases break down phosphoric acid esters. Then proteases. So as you know, proteases, so these are enzymes that act upon protein, or they break down protein molecules into polypeptides, and again further it leads, further breakdown leads to formation of simple amino acids. So these are proteolytic enzymes, means they act on protein or they break down proteins and hydrolyzes protein into simpler components such as proteose, peptone, and amino acids. Okay. So these enzymes are involved in hydrolysis of milk protein, just like casein and whey proteins. So proteases, they mostly in milk they act on casein as well as whey protein. So proteases enzymes, they get destroyed at temperature around 80 degrees Celsius. Okay. So proteases, after heating the milk at 80 degrees Celsius, they get destroyed. The next, reductase. These enzymes help with reduction of certain organic compounds. They reduce methylene blue added to milk and make it colorless. So reductase we mostly use in methylene blue test as an indicator. Okay, so the enzymes basically it helps in reduction of certain organic compounds. Then next important enzyme is lactases. So as this name suggests, lactases, these are the category of enzymes. They hydrolyze lactose into glucose and galactose. So lactose enzyme basically act on sugar lactose. That lactose sugar is disaccharide, which is made up of glucose as well as galactose, and after breaking down, that lactose gets split into glucose as well as galactose. So lactases, these are the enzymes, they break down or they bring about breakdown of lactose sugar present in milk. The principal enzyme is beta glycosidase. So the main enzyme present in milk in the lactase category, it is known as beta galactosidase. The above enzyme hydrolyzes lactose, which is finally converted into lactic acid. Okay. So in this particular lecture we have seen different types of enzymes and their role. So in next lecture we are going to study milk carbohydrate. Okay, thank you.